Hello students, once again I welcome you all to Misostudy. So we are discussing some questions which are important as per your NEET is considered, and we are discussing about the chapter strategies for the enhancement of food production. In total I am going to discuss total 10 questions. Those 10 questions I have chosen from different part of this particular chapter. Some are with the help of a plant tissue culture and second from the animal husbandry. In the whole chapter or the session, we have discussed how we can improve the plant product, even how we can improve our livestock, that we have already studied. Now I will discuss the questions side by side. My motive of taking such 10 questions is that these 10 questions, first of all, they are important. I am not saying they will directly come into a NEET exam. I am just saying that the such type of questions will be asked in the next NEET questions. So let us discuss with the first question. Choose a set of the bacterial disease in poultry. As they are asking about some of the disease which is because of the bacteria, and I have to choose the two important bacterial disease as per the poultry animals they are considered. When we talk about the first option, out of these first option, the ranicate, this is a viral disease. This is a viral disease. So this option is wrong, right? We have to look for the both in which they should be bacterial. This is a bacterial disease, whereas this is also a bacterial disease. This is also a viral disease. We know that. Let's look for the C option, fowl cholera and infectious coryzae. These are what? These both are bacteria. So, the option 3 is correct one. Similarly, when we talk about the D option, this is because of the fungi. This is because of the fungi. Now, let's read the question again. They said that we have to choose the correct option which is because of the disease, which is because of the bacteria. Ranicate and the mark, these are nothing, these are just a viral diseases, but we are considering here only bacterial diseases. Similarly, the question, option B, ranicate and the fowl cholera, this is a bacterial, whereas that is a viral one. In the C option, both of these, they are bacterial disease, aflatoxis, this is a fungi disease. So the correct option for the question number 1 is a C option is a correct one, right? We have already discussed about what are the problems which are associated with the poultry. So this is how they can ask some questions from this chapter only. So this was the first question. Hope this first question is clear to you. Let's move on to next question. By the time, let me prepare board for you. Now, read the question. Fish selected for culture practices must possess some feature to make culture profitable and identify the wrong one. Means what we are going for? We are going for the culturing of fish. That means what we want a fish in a bulk amount, and we know that why fish is important for? Fish is important for the protein. Let's look for the option, then we will judge that which one is the right and which one is not right. A, fast growth rate. Yes, the fishes, they should have a fast growth rate. This is important. They should be there when we talk about the fish culture. When there is a fast growth rate is there, that means healthy offsprings they will be produced, healthy fingerlings they will be produced, and heavy or you can say a good quality fishes will be produced that will be having a good amount of the protein. Late maturity. Late maturity, is that reason? No, not at all. Because if they will mature later in their life, there are chances that the reproduction rate will be low. So because of the late maturity, reproduction rate will be low. When reproduction rate is low, in that cases the number of the fingerlings which are produced will be less. So less fingerlings they will be produced, less will be produced, we will not be able to culture properly, we will not be able to get that much amount of you can say the fishes which we are looking for. Similarly, the C indicate the disease resistance. Yes, our fishes should be disease resistance. Like in our case, we are exposed to bacterial diseases, we are exposed to viral diseases. Similarly, in the case of fishes also, the bacterial diseases are there, the viral diseases are there. So, they should be resistant to such type of bacterial as well as viral diseases. Next is the high nutritious value. Yes, more nutritious value, that means more the culture should be the culture will be healthy, that means more economic that particular culture will be. So, the correct option that it should not be there, that is a late maturity. We have to look for those fishes which mature at an early stage. So, the correct option is a B, late maturity. Now, Hisar Dale is obtained by crossing of. A very common question, very easy and a direct question. No need to go into the in here and there. The correct answer is Bikaneri ewes with merino rams. Now, in this question, one twist is there. Students might get confused regarding the option B and the D. Can you see this option is somehow similar to the option D? Yes, they are similar, but the difference is that instead of merino ewes written over here, the merino rams are there. So, this is quite confusing. So, students, whenever you are reading something, just write down side by side. Just write it once, twice, thrice, then only you will be able to learn all those things. Otherwise, once you have read this, okay, this is the case, and you know that because of the hybridization between the Bikaneri and the merino and Hisar Dales, they are produced. No, this should not be the case. You should write down each and everything once, twice and read that again and again and again. I am telling you, because whenever you will slip from a single word, you will be negative with 4 marks. So, this was for the question number 3, the correct option is a D. Now, let us talk about the next question. In which method of animal breeding, now they are discussing about the animal breeding. In which method of animal breeding the 2 different species of the male and the female animals, they are mated. Now, what are they saying? They are saying they have 2 species, species A and species B. We have 2 species. Out of these 2 species, they said that in which of the methods animal breeding, the 2 different species, one of female or the male are the mated. Suppose I have male for species 1 and a female for species 2. What I do, I prepare one breeding experiment and what I will get is a hybrid. Hybrid of what? Hybrid of A and B. Why we go for this hybridization experiment? So that whatever the good qualities which is there in the species A, whatever the good qualities, whatever the good gene which is there in the species B, they should come in that hybrid. So, this is the major reason why we go for a hybridization experiment. In this experiment, because we are dealing with the 2 different species, species A and the species B. Now, what is the name that can be given to such type of hybridization? That is called as interspecific hybridization. Interspecific hybridization, that means between the member of 2 different species. When we talk about the hybridization in case of the members of a same species, this means members of different species, and when we talk about the hybridization between the members of same species, that is called as intraspecific hybridization. That means this is a type of hybridization between members of same species. members of same species. This is called as a intraspecific hybridization. Now come back to the question. They are asking about the two different species of males and the females. That directly indicate that this is a interspecific hybridization, right? Correct option is option number B, interspecific hybridization. So hope the two words inter and the intra is clear to you. Now let's talk about the another question, fifth question. This was asked in NEET 2016, I guess, this question. Now let's see. A cell at a telophase stage is observed by the student in a plant brought from the field. He calls his teacher that this cell is not like that of other cells at the telophase stage. There is no formation of a cell plate and thus the cell is containing more number of chromosome as compared to the other dividing cells. This would be. Now what he saw that the cell plate was not there. That means in this cases the cytokinesis is not happening. Another important reason or another important thing was that they said that he observed that they were having a more number of the chromosome. Now come at this side. We are discussing about a cell and that cell is at telophase. Sorry. He discovered that this particular cell he has studied. He said that in that cell he saw that the number of chromosome they were comparatively higher as compared to the another cell which is having different chromosome, or as compared to the another cell which is having at the telophase stage. When we talk about the cell, when cell has to divide, it divides by two processes. One is called as a karyokinesis and another is called as a cytokinesis. Karyokinesis means division of the nucleus. That means whatever which is present inside the nucleus, either the DNA that will replicate and that will divide. That means the chromosome number they will double itself and later on they will migrate towards each and every cell. Now they said that cytokinesis is not happening. That means cell plate formation is not happened. Cell plate formation is not happened. That means that cell will be having the double number of the chromosome, double, more than double, 3 times, 4 times, 5 times, 6 times number of the chromosome they have, because that cell has not undergone the cytokinesis. The cell plate formation has not taken place. So what has happened? Instead of having a single set of the chromosome or the 2 set of the chromosome, if it is a diploid, then there are chances that the 3 set of the chromosomes they are present, the 4 set may be present, 5 set or 6 set may be present. This is because the cytokinesis has not happened. In that cases, when the chromosome number is more than 2 set, that is called as, condition is called as a polyploidy. That is because of called as a polyploidy. Now, so the correct option is a polyploidy. That means having more than 2 set of chromosomes. More than 2 set of the chromosome, right? That means the cell has not divided yet. So this is condition is called as a polyploidy. Let's talk about in case of the wheat. For example, wheat. Scientific name Triticum aestivum, and in this case we can see that a hexaploid condition is seen. That is 6x or 6n, whatever you want you can write. That is a 6 set of that particular chromosome is present. That is in case of the wheat Triticum aestivum. So this was the 5th question. The correct answer is option number A, called as a polyploidy. Hope this question is clear to you. Let's talk about the next question. Question number 6. Now, this question states to obtain virus-free healthy plants from a deceased one by tissue culture techniques, which part or the parts of the deceased plant will be taken? Now we have to go for the plant tissue culture experiment. In that cases, if a plant is deceased one, we go for the apical meristem only. Now, why we go for apical meristem? The reason is that they act as interferon. Now, why I am calling it as an interferon? It's just that the covering which is present surrounding it, they do not allow the pathogen to enter into it. Have strong covering outside the cells layer, which do not allow pathogen to enter, which do not allow the pathogen to enter. This is the reason why we always go for apical meristem only. So, for the question number 6, the correct option is A. Next we have is the 7th question. In a plant breeding programs, entire collection of plant trees having all the diverse alleles for all the gene in a given crop is called as. This was the whole method of producing any kind of new recombinant, you can say, plant. In that condition, what we go for, first of all, the first condition that I have told you, that is a collection of the germplasm. Now, what is germplasm? That means we have to collect all the desired, not only the desired, even those alleles of that particular varieties. That is called as their germplasm. In these cases, it is written over here, that means we have to collect entire collection of all the diverse alleles of a gene, if a given crop. That means either these particular genes, they are in a one form, or you can say some wild varieties are there. Some are which are developed by the breeder also. Likewise, we have studied the different alleles they are there, and these varieties, some breeders and some commonly available, some which are having a high yield, some provide disease resistance, all the different alleles they are collected. Later on, out of those different alleles, we only look for our desired gene. That is called as a germplasm. The whole collection that we go for before going for a hybridization experiment, that is called as a germplasm collection. So, the correct option for the 7th question is a C. A indicate cross hybridization among the selected parent. No, this will be done only after the collection of all the desired alleles. Evaluation and the selection of the parents, this will be done only after the selection of the germplasm, and selection of the superior recombinant, this is also not the part. So, for the question number 7, the correct option is a germplasm collection. Hope the things are clear to you. Now, Wonder wheat is a new wheat variety developed by. Let's discuss about the wonder wheat first. What is wonder wheat? Wonder wheat is that wheat whose yield is around 18 tons per hectare. 18 tons per hectare. And one more important thing that I would like to discuss about the wonder wheat is that when we see the number of grains per stalk, that is also very high. That is around 200 grains per stalk. This is also a very good number. The whole two number, they are very good number as per the Green Revolution is considered. So, the wonder wheat is a very important wheat. It was first of all developed by the Mexico International Wheat and the Maize Improvement Center, right? This you have to remember. That's for the question number 8, the Wonder Wheat. This is developed by the Mexico's International Wheat and Maize Improvement Center. Correct option A. As per the question, 8 is considered. Now, let's talk about 9th question. By which of the following methods new and the better varieties of the plant can be obtained? Now, let's understand this. Suppose I have one variety. This is a variety A and this is a variety B. If I say about the variety A, this particular variety, this is having high yield. They are having high yield is there, whereas no insect resistance is there. No insect resistance is there. Whereas the variety B, I can if I say in these cases, if in this cases, suppose in variety B, the low yield is there and have insect resistance. Insect resistance is there. What I want that in the hybrid, in the future generation, what I want that particular type of product in that high yield should be there. High yield should be there. Simultaneously, there should be insect resistance. Now, how will I proceed? I will proceed because from variety 1 I need this, from variety 2 I need this. Now how will I proceed? I will bring about the hybridization experiment between the variety A and the variety B. Suppose the 1000 number of the seeds they are produced. 1000 number of the seeds they are produced. Out of these 1000 I have to select desired character. I have to select a desired character, that which of the following seed our desired character, that means high resistance and high yield is there. I will look for, suppose out of these only one will be having our desired character. Out of these one, what I will make, I will make a copy of these. And producing the copy of it is very easy by self-pollination process. I can produce a copy of it. Now what I have done? First of all, I have planned one hybridization between these. This is what? Hybridization. And after hybridization, what I have done is a selection of a desired character. That means I have selected those desired character which I am looking for. So this is the whole basis of, you can say, the hybridization experiment, first hybridization and second selection. Now come back to the question. By which of the following methods new and the better varieties of the plant can be performed? Yes, selection is a method, but they should be followed by the hybridization. First, what we have to go for? Selection of the germplasm, collection of the germplasm, then selection of the parents, then hybridization experiment, and then selection. This is also a right option, this is a wrong option. But when we talk about the option D, which indicate hybridization followed by the selection, this seems to be more correct and more precise. So, while performing any of the question, while attempting any of the question, do this thing in mind. mind must read all the question as per this particular, is considered when you have only 4 options. I am not saying in that cases when you have a column 1 and the column 2, in that cases you can skip one option, because if you are getting an answer from the 3 points and you can see a desirable change within the option 1 and 2 and the 3, you can go for, you can go to the next question without reading the next option. But in this case, don't go for any other option. Otherwise, if I am in a hurry, what I'll do, I'll read the question, what I'll do, yes, selection is the right one, that after the selection that I'll get the desired. Or sometimes some of the students will write hybridization. They'll go to, okay, hybridization, this is the right hybridization occur. But out of these, the most correct is a hybridization followed by the selection. This is how you can proceed. Correct option for the question number 9 is the D option. Now, the name Norman Borlaug is associated with. The Norman E. Borlaug. Norman E. Borlaug, N.E. Borlaug, is associated majorly with Green Revolution. Right? Very common name, very easy question, and this was asked in some of previous year NEET exam. So the correct option is option number A. Now this is a universal one. Now tell me one thing, who is the father of Indian Green Revolution? Father of Indian Green Revolution. Who is the father? Father is M.S. Swaminathan. M.S. Swaminathan. This is also very important. M.S. Swaminathan is father of Indian Green Revolution. M.S. Swaminathan. So the correct option for the question number 10 is a Green Revolution, option number A. Hope students, all of these questions, the 10 questions, they are clear to you. Don't stop here. Practice more and more questions. More you will practice, more you will attain success very fast. Because NEET, the next year NEET exam is not very far, it's near only. You have only countable days. Plan accordingly, revise the questions again and again and again. Watch lectures again and again. Make notes by yourself with your own pen and paper. Write down everything, read that again and again. If you are not able to learn anything, just read that again and again. Prepare cheats for that. I'm not saying the prepare cheats, I'm not saying that. I'm saying prepare cheats how you can remember that. Any place where you go frequently, like your table, you used to, suppose you used to sit on the chair and the table, what you can do, make a separate chart of it, stick that particular chart. Suppose you are not able to remember these fathers, you are not able to remember some of the important scientists, what you can do, make a chart, stick over there, read that again and again and again. I'm sure within 1 or 2 days you will be thorough with all those things. Nothing is impossible, even impossible says I am possible. So thank you so much students for watching this. We'll meet in next session.